Hi everybody, this is Dr. A and our third and last video on microscopes, part of the Labware series. We are going to look at all the different types of microscopes that are out there that can be used. So we're going to start with the most commonly used, which is the Brightfield microscope. It's a compound microscope. It is the primary microscope used in the clinical laboratory and in your student labs. When you work with that microscope, you will see a light background on your specimen and usually a darker image. Often slides have to be stained so they are visible, so the components are visible. Next is the Darkfield. So it is used in the clinical lab to observe spirochetes and exudates, for example. It uses a special substage condenser that causes the light waves to cross on the specimen at oblique angles and not parallel like the Brightfield microscope. So it goes oblique like that, and only the light that is scattered by the specimen can reach the objective lens. And so in a darkfield microscope, you have a dark background with a light image. Next, we have the Differential Interference-Contrast microscope. So this one will give you more of a 3D image of the object. So it is a Brightfield microscope that can be modified into a differential interference-contrast microscope by adding a special beam splitting prism to the condenser. So the two split beams are polarized. So one passes through the specimen, and that alters the height of the light waves. And the other is a reference beam, and it does not pass through the specimen, but then both beams pass through the objective lens separately and are recombined by another prism, which is what gives it the 3D image quality of it. So this is great to show fine details in urine sediments without using any kind of special staining. Next, we have the electron microscope. So it functions with the same general principle as the light microscope, but the electrons that are traveling in a stream, they have the same wave properties as light beams, and so we can capitalize on that in the electron microscope. So the limit of magnification with light is between 1,500 to 2,000 times magnified before the resolving power is decreased and lost. With the electron microscope, though, we can keep good resolution up to 50,000 magnification. So there are a couple of types of electron microscopes. We have the transmission electron microscope. So the electrons are accelerated by a high voltage potential, and they pass through a condenser lens system that's made of two magnetic lenses. And then the electron beam is concentrated onto the specimen, and then the objective lens provides the primary magnification. And the final image cannot be viewed directly, so it's not like you're using your eyes to look into oculars like you would with a Brightfield microscope, but it's the image that is projected onto a screen or a photographic plate. And then you have a variation of that. It's the scanning electron microscope, and so it focuses the electron on the surface of the specimen, not through the specimen, right? And it produces a 3D image. So you can see here the electron microscope image here has more of a 3D quality to it. And so the beam of electrons will strike the sample, and the electrons are emitted from the surface of the sample, and the electrons that are deflected from the electron beam then are focused onto a cathode ray tube or a photographic plate, and that generates the three-dimensional image. This is what a scanning electron microscope looks like. So in both electron microscopes, the specimen does need special preparation, and so this is not done in routine clinical labs. This is more done in research. The specimen must be very thin for electrons to pass through, because the electrons have very little penetrating power. So that's for the transmission one. The electrons have to transmit through. But in the scanning one, the specimen can be thicker because the electrons don't have to pass through the specimen. They're actually bombarded onto it and bounce off of it. So you can't use the electron microscope to study living cells. So if you want to study motility or something like that, because the cells get damaged too easily by the process. but you can get really detailed images, for example, of cell structures. So this is an example of the type of image you can get, where this is an image of a mitochondria from within the cell. And so you can see the membranes of mitochondria, the cristae, and everything. So quite a lot of detail there. Next, we have the fluorescence microscope. So fluorescence involves the excitation of a compound with a high energy wavelength of light. So it's usually a blue violet or ultraviolet wavelength of light, and then that causes the compound to emit a lower energy wavelength, which is usually a green, red, or a yellow. These fluorescent compounds, they can be coupled with antibodies or DNA or RNA probes, and so they're used in a lot of immunoassays, but also you can do this in microbiology, tagging antibodies with fluorescent compounds and then using those antibodies to detect specific types of bacteria. Then the ones with DNA and RNA probes that are tagged with fluorescence, that's what's used in like microarrays and molecular diagnostics. So the fluorescent microscope is basically a dark field microscope with a wavelength selector. The dark field condenser then is preceded by a special exciter filter that only allows the short wavelength of the blue light to pass and cross on the specimen plate, so that's that high energy wavelength. And then the fluorescent compound in the specimen, if it is present, then will absorb the blue light and then emit a yellow or a green light. And so you have to put another special filter on the microscope on the tube or the eyepiece, and it only allows the emitted light of that desired wavelength to reach the eye to be seen. So it only allows green or only allows the yellow to be seen. And so you don't see the blue light that's being shone through onto the sample. And so basically you're going to look through the microscope and you'll either detect fluorescence, as seen here, or none at all. It'll be just dark. So this is used in the clinical lab, especially in microbiology. So fluorescence staining techniques or fluorescent techniques do have more sensitivity than traditional serology and can be used on microbes that can't be cultured also, so that's really nice. And it's also widely used in cellular and molecular biology. Next, we have a phase contrast microscope. So this microscope uses a hollow cone of light instead of a solid cone of light, like in a traditional Brightfield microscope. And this cone of light has to be aligned with special rings in the objective lenses to produce the best contrast. The principle is that the light slows down as it passes through the specimen, and the phase rings in the objective lenses can change the phase of the light, converting the lower light speed to an intensity that increases the contrast of the specimen. So it's a Brightfield microscope with a modified objective lens and a modified condenser. It requires additional alignment steps to work, so a little bit more technical knowledge to make it work. And it slows down the speed of light by 1/4 of a wavelength, which makes it sensitive to the differences in refractive index and causes that contrast to be apparent. This is useful for observing cellular structures inside of cells and cast in wet mounts like of urine, or observe vaginal smears where you would look for, cells in, maybe trichomonas or something like that. And also you can use it for counting platelets. And then we have a polarized or compensated polarized microscope. So it is another adaptation of the Brightfield microscope. So in this one, as light passes through the specimen, some objects, such as crystals, can bend the light, so it just rotates the light. And so if you add a filter above the specimen and it's at a different orientation from a filter below the specimen, the light that has not been rotated or polarized by the specimen cannot pass through the top filter. It can't be detected. So it's dark. You can't see anything. Okay. So the polarizing filters only allow light waves of one direction to pass through. So if you look at it here in this diagram, you have a light source, and then light waves are on in all different orientations. Okay. But then if you put a polarizing filter, it will only let the light waves that are parallel to the polarizing filter through. Okay, and so that is polarized light. So in a polarizing microscope with a polarized filter, which is placed between the light source and the specimen, and then another one is placed above the specimen between the objective and the eyepiece, one of the polarizers is going to be rotated until they're at right angles. So if they're at right angles to each other, then light is completely blocked, right? And so the viewing field will be completely blacked out because, you know, both wavelengths, both orientation of the wavelengths are blocked, okay? So any object that can rotate or polarize light it can be seen as a light object against a dark background. So it's overcoming the fact that the filters are perpendicular to each other by bending the light and making it parallel to that top filter where you can actually see it. And then there is also a compensated polarized microscope, and so in that one there's a red filter also that's placed between the two polarizing filters. It's placed at a 45-degree angle, and so it makes the background magenta instead of black, and the polarized objects are yellow or blue. So this is excellent for crystal analysis in synovial fluids where you're trying to distinguish monosodium urate from calcium pyrophosphate. And so one is blue, the other is yellow, and it depends on the orientation of the polarizing filters and stuff. And then lastly, a little note on digital microscopy. So more and more you're seeing combining of digital imaging plus the artificial neural networks. And those are used to, for example, automate manual differential when we're doing cell counts and stuff. And, you know, in a differential, you differentiate between the different types of cells, but it goes beyond what a cell counter does. And so the artificial neural networks are a type of artificial intelligence that tries to mimic human thinking. And so they've been used increasingly in medical imaging to scan through CT scans and radiography scans and MRIs, etc. And in medical imaging, they're using this in addition to a radiologist reviewing the scans. And so these neural networks are looking for patterns, looking for things that could be, you know, significant. And so with digital microscopy, you can have virtual microscopy, so you can have images that can be shared and sent. Of course, in a traditional microscope, you can take pictures of images, but if a tech does a diff on a regular microscope, you know, other than the notes that they make, there's really no record of it. Somebody else would have to pull the slide and diff it again, and maybe to see what the same thing, or maybe they don't. And so with the virtual microscopy that can be done in images that can be shared, there can be more collaboration. This can save time and workforce energy. And the cells are pre-classified by types, usually through the digital microscopy. And so that's one of the helpful things in that. And then one of the ones that is out there is called a Cellovision. So there you go. That's where it's going. And I hope this gives you a good little overview of all the different types of microscopes that are out there. So anyway, stay tuned. I'll be posting some more soon.